My name is Saul Schaefer. I'm a professor of medicine at the University of California, Davis in California. And I recently presented my talk entitled Silencing of the Cardiac 18 kilodalton translocator protein Prevents Heart Failure Due to Pressure Overload. This was presented at the International Academy of Cardiology Annual Scientific Sessions 2018 in Boston. As background for my presentation, we and others have shown that heart failure is in part due to energetic failure, that is, a reduction in the production of adenosine triphosphate, ATP, and the consequent increase in reactive oxygen species and damage to myocytes, causing left ventricular dysfunction and fibrosis. We've also previously shown that the level of mitochondrial calcium and the uptake of calcium into mitochondria prevents damage due to ischemia reperfusion and may be involved in the pathogenesis of heart failure. Our prior studies have also shown that the 18 kilodalton translocator protein, also known as TSPO, can modulate the entry of calcium into mitochondria through the voltage-dependent anion channel, or VDAC. Based on these studies, we postulated that first, heart failure increases the expression of the translocator protein as a response to pressure overload stress, and consequently that silencing of the translocator protein expression would reduce TSPO and allow normal mitochondrial calcium uptake. In order to test this hypothesis, we used an animal model of pressure overload caused by transverse aortic constriction, or TAC. In these TAC animals, 8 weeks of pressure overload results in heart failure manifest primarily by a reduction in ejection fraction of approximately 50%. In the animals in which the translocator protein expression is silenced through a conditional knockout, we have reduced ejection fraction, in the animals in which there's genetic silencing of the translocator protein, ejection fraction is reduced by approximately 10% rather than the 50% reduction seen in the wild-type animals. In order to determine whether this was due to a change in mitochondrial calcium uptake, we measured calcium uptake under different external calcium loading conditions using permeabilized myocytes from both wild-type and knockout mice. In these experiments, we showed that calcium uptake was reduced in the heart failure mice under wild-type conditions and normalized, returned to normal, in those with the conditional knockout. In concert with this normalization of calcium uptake, we also demonstrated normalization of ATP production, such that ATP production, especially with complex 1 substrates, was restored to normal. Concurrent with the normalization of ejection fraction over the 8-week period, we showed lower levels of atrial natriuretic peptide, less clinical manifestation of heart failure, and relative normalization of cardiac ultrastructure based on mitochondrial size and density, as well as decrease in collagen production in the conditional knockout mice. Since we hypothesized that normalization of energetics would result in decreased reactive oxygen species, we measured ROS production, and that was also decreased in the conditional knockout mice. Finally, we measured mitophagy and saw that mitophagy was increased, resulting in more normal mitochondria in the conditional knockout mice. In summary, we've demonstrated that TSPO, the 18 kilodalton translocator protein, is upregulated in heart failure, and that genetic silencing of this protein expression normalizes the response to transverse aortic constriction and may point the way to a clinical intervention to prevent or treat heart failure in man.